Hi everyone, this is Kevin Wagner, the Keto Advocate. In January of 2016, I had the privilege to sit down with Dr. Stephen Cunanne at the first annual conference on nutritional ketosis and metabolic therapeutics held in Tampa, Florida. Let's see what Dr. Cunanne had to say.
I'm a professor at the University of Sherbrooke in Quebec. I have a PhD in physiology. I taught nutrition for 18 years at the University of Toronto, and I'm now doing more work on brain energy metabolism and nutrition in aging.
Okay. Could you tell us about the topic you'll be speaking about here at the conference?
I'm going to talk about brain imaging, how to image the way the brain uses its fuel as we get older, and the implications for understanding the origin of Alzheimer's disease and potentially the treatment of Alzheimer's disease. So we're studying the way the brain uses fuel as it gets older. We need to use PET imaging to study brain fuel uptake, and the classical way of doing that is with the glucose tracer, so you can look at the way the brain uses its sugar. We're lucky in having the only lab in the world at the moment that is actually measuring brain ketone uptake as well. So this allows us to examine how the brain uses those two fuels as it gets older, as it starts to confront diseases like Alzheimer's disease, how the older person who might still be healthy, how their brain energy metabolism changes, and how this can change in relation to some kind of intervention that you do, some kind of experimental treatment, whether it's a ketogenic diet, whether it's a ketogenic supplement, whether it's exercise, or other treatments. Could be a drug treatment as well. So we get a chance to see how the brain's responding using this brain imaging technique. Glucose metabolism goes down in the older persons, in the brain of the older person, that's for sure. It's been reported previously, but I think what was never really clear is whether those individuals were really screened to establish whether they were cognitively normal. Because the problem is that as we get older, we're also more susceptible to memory problems and cognitive decline. And so if you say what's happening during aging, are you trying to distinguish healthy aging or the aging as you slip towards these cognitive problems? And so to be sure that it's aging, you have to show that there are no cognitive problems. And when we've done that, we'll be reporting that at the meeting. And we've got over 40 individuals in that group, which makes it a fairly solid set of results. And it shows very clearly that there's about a 10% decrease in the amount of glucose that gets into the frontal cortex, so the frontal part of the brain. And that it seems to be a universal observation that corroborates 20, 30 years of work in this field. And I just think it's been done in a more robust way than perhaps has been done in the past.
Is that area of the brain in particular significant for cognitive function?
Well, it's the area that's called where we make the executive decisions, so where we analyze how to attack a problem. It's the problem-solving sort of area in particular. It's not considered to be the area that really controls memory, but it's part of decision-making, which is a big part of using your memory in a sense. So yes, it's really important. Well, the imaging technique called PET imaging, positron emission tomography, shows the amount of uptake of glucose into the brain. And you can do this in two ways. You can do it in a qualitative way, which means you can show that there's a difference between two people or a difference between two groups of people, but you don't know exactly how much that is. It's qualitative. Or you can use a bit more involved approach where you have blood samples and you show exactly how much of the radio tracer is getting into the brain at the level of the artery getting into the brain, the carotid artery. You don't take the blood sample at that level, but that's needed in order to calculate the exact amount, the magnitude of the difference. So you can say it's 10% lower in this individual in this area, or it's 10% lower in this group globally in the brain. And that's hard to do unless, it's impossible to do unless you have that quantitative approach, which is a bit more involved. It's a bit more demanding for the patient, but it's much more useful in trying to evaluate how things are changing and how much they've changed. Because brain imaging today uses a lot of statistical mapping methods to show differences that are statistically different between two areas, but you don't actually know how much the difference really is. And to us, that's important if we're going to think about how we're going to change a deficit that's in one area. You need to know what the deficit is, and you need to know then how much you need to provide of your treatment that's going to change that. So in order to answer that, we need to study people who are at risk of Alzheimer's disease but don't have it. So there's genetic populations that are like that. There are people that have a family history, especially on the maternal side, that have a family history of Alzheimer's disease. Aging itself, even when you're still cognitively intact, is a risk factor for it. Insulin resistance is a risk factor for it. Diabetes is a risk factor for Alzheimer's disease. And in each of those cases, you can evaluate whether the person has a cognitive deficit or not. If they don't have a cognitive deficit, then they're at risk of the disease, but they don't have it clinically yet. So then you can measure their glucose uptake in the brain, and you can say, okay, this person is at risk of Alzheimer's, but they don't have it. Do they have a deficit in glucose uptake or not? And the answer in those 5 cases is they do. In fact, they can be as young as 20 years old and have that deficit in glucose uptake, but they're still cognitively normal. They're still functioning as they would be expected if they didn't have that risk factor. So yes, glucose problems in the brain can result from the disease, from Alzheimer's disease, but they can also contribute to it. So it's what we think of as a vicious cycle. And we're not saying it starts with the glucose problem, but we're saying that the glucose problem comes long before the actual clinical problem, the problem with memory and cognition. So the PET scanner will indeed show that the glucose uptake is getting to the brain cell, but it doesn't say what the brain cell is doing with the glucose afterwards. And that step, as you mentioned, is called glycolysis. So all we know with the PET scanner is that the glucose is not getting into the brain as it should be. Now, normally what controls the rate that the glucose gets into the brain is the rate of activity of the brain cell itself. So the brain cell sends a message to its neighbor, and now it's used some glucose to do that. So its transporters are turned on and it pulls more glucose into itself, and then it can send the next message and the next message and so on. So if less is getting into the cell, it means the cell is not working very effectively. We don't know for sure that the problem is with glycolysis through the PET scanner, but we do know that the brain cell is not working well, and that's why it's taking up less glucose. So you have to do a different type of study to show that the utilization of glucose, the glycolysis part, is affected. And people have done that with samples of brain that have been obtained from people who died with Alzheimer's disease, and they measure the activity of those steps in the way that glucose is converted into energy, the glycolysis steps. And with that type of approach, you can show that glycolysis is blocked or is impaired in Alzheimer's disease. So the pattern during normal aging is to have lower glucose uptake in the frontal cortex, the front part of the brain, and almost no other area seems to be significantly affected. A person of the same age who has Alzheimer's disease has got a deficit in the frontal cortex as well, but it's a much more severe deficit in the parietal and temporal cortex, this area above the ears. So the aging brain, we don't see that defect at all. And the Alzheimer's brain, it includes the frontal cortex, but it's spread to the temporal and parietal cortex. So it's a different pattern. It's a more severe pattern. The magnitude of the effect is greater, and the areas are larger. There's more areas affected. So what we've been able to do with ketone PET is to show that neither aging nor Alzheimer's disease affects the uptake of ketones by the brain. at least up to a stage what they call moderate Alzheimer's. We haven't studied anyone with severe Alzheimer's yet. So this suggests that the neurons, the brain cells, can use ketones when they're provided, but they can't use glucose because glucose is not getting in. They probably could use glucose, but there's problems with glycolysis and there's problems with glucose transport. So ketogenic interventions should have potential benefit in Alzheimer's, and indeed that's been demonstrated in a number of studies already clinically. So we've got a sort of mechanism where we've got proof that the brain can use ketones, and it would be reasonable to expect to see the clinical results that have been reported. The question is now to optimize that. When do you start? What dose do you give? Will people tolerate ketogenic treatments for extended periods? Because once you've got memory problems, it's not like solving a cold or an infection. It's something that's going to be around for a long time. It could be. I can't speak with authority to that question, but I can say that ketones require mitochondria for them to be utilized by the brain. So if brain ketone metabolism is normal in Alzheimer's disease, at least in the mild to moderate stage, the mitochondria must be able to do most of their work normally. That doesn't mean there aren't defective mitochondria, but the capacity of mitochondria to use ketones still seems to be relatively normal. So there may be problems, and other people have cited evidence for problems with the way mitochondria function in Alzheimer's disease. It's certainly not advantageous that they would have a problem, but it looks like it's not necessarily limiting them the ability to use ketones, which would be critical in bypassing problems with glucose. Yes, there is. This has been studied. Before we had PET imaging of either glucose or ketones, people were studying the way the brain used glucose and ketones. Ketones are actually the preferred fuel of the brain. So when they're available, they go in first. They get priority. And what happens, whether you do a glucose infusion into the blood, whether you do a ketone infusion into the blood or put people on a ketogenic diet, we'll be reporting those results at the meeting today. Glucose uptake will go down in relation to the amount of ketones that are being provided, so that the total amount of fuel that the brain gets is still optimal. If one piled on top of the other, you'd have excess fuel and excess stress, in fact, on the mitochondria, and that's not advantageous. So glucose uptake is dependent on the activity and the requirement of the brain cell for fuel. And if ketones are supplying part of that, then the uptake of glucose goes down in consequence. And that's a totally normal reaction. Usually what happens is the glucose is the one that isn't available, because ketones are the preferred fuel, but they're not the dominant fuel in terms of amount. So if you've got less glucose because you've been fasting for 3 days or 4 days or a week, then ketones are being produced and they're compensating for low glucose. But if you turn it around and put people on a ketogenic diet, then the ketones are available in excess, and the glucose uptake will go down in the brain. And we've confirmed that using the PET imaging with both tracers. It's a hard question to answer. The brain can use all three, so beta-hydroxybutyrate, acetoacetate, and acetone. Acetone has in fact been used in treatments for epilepsy, in experimental treatments in animals. We don't know how it's metabolized by the brain. We do know that acetoacetate is the fuel that gets in, of the two ketones, beta-hydroxybutyrate and acetoacetate. It's acetoacetate that is burned by the mitochondria, so burned by the furnace of the brain. So beta-hydroxybutyrate is a stable kind of transport form of ketones, but it's not utilized by the cell. It's got to be converted to acetoacetate, which is an equilibrium that is going on all the time in the body. So the conversion is always happening back and forth, and you buffer some of the acetoacetate and convert it to beta-hydroxybutyrate to keep it, to get it around the body, to get it where it needs to go. But as soon as it's at the destination, then it's converted to acetoacetate and is burned that way. Well, we know that it can have beneficial effects in certain epilepsy models. So there's it must be doing something. It can also, surprisingly, be converted to glucose. A certain amount of it can be converted to glucose. At least that's been shown in human studies. So it's possible that it's being metabolized by ways that we don't currently study. Those studies were done in the 60s, and I don't think anyone's followed up on them. In fact, Hans Krebs, the founder of the Krebs cycle, Nobel Prize winner, one of his main interests was ketone metabolism, and he studied acetone metabolism. So we don't know a lot more since that period, though, and it's perhaps there's some surprising things that we would learn by getting back to basics. So ketones are important for early brain development for two very important reasons. One is that the brain is very big in an infant compared to its size, so it's consuming a huge amount of energy. In adults, it's consuming 2% of our energy. Sorry, it weighs about 2% of our body weight, but it consumes about 20 to 25% of the energy consumed by the body. In the infant, it's 75% of the energy utilization of the body goes into the brain, which weighs 11 to 12% of its body weight. So it's much bigger proportionally. That's the first point. The second point is that glucose, there doesn't seem to be enough glucose available in the infant's when it's feeding and in its ability to make glucose by gluconeogenesis to supply the brain's needs. So ketones aren't an alternative fuel, they're an essential fuel for the infant's brain. And the third point is that ketones do more than make fuel, they actually are the main substrate to make the lipids of the brain. Because cells have proteins in them, but they have a lot of lipids. Cholesterol is an extremely important lipid in the brain, and almost all of it is made in the brain from ketones. Most of the saturated fats in the brain, the palmitic acid, stearic acid, oleic acid, are made from ketones. So ketones are doing two major, they're sort of anabolic in terms of building the structure of the brain, and they're catabolic in terms of supplying fuel for the brain in making up for an inadequate amount of glucose in the infant's body, the mother's milk. So there are MCT, the equivalent of MCT, medium chain triglycerides, in milk, in the milk of all mammals, in fact. So all mammals have evolved with the ability to use ketones while they're infants. And when they're weaned, those medium chain fatty acids are no longer available, except in humans who have body fat when they're born, a term, if they're born at term, and some of the medium chain fatty acids that are in milk are actually stored in the fat. So it kind of prolongs lactation, even when lactation is finished. In terms of supplying ketones to the brain, human infants are unique in having a longer term supply, which I think is actually how we evolved as humans in the first place. So survival of our fat babies made us human in prehistoric times. Well, the short answer is we don't know for sure. There are data that show that you can get a bigger ketone punch out of C8 than out of other MCT, but that's the net ketone production. That's the combination of acetoacetate and beta-hydroxybutyrate. Is that true in older adults? Is it true in Alzheimer's disease? And does it matter? Is it the total amount of ketones that are being produced that's the most important factor, or is it the ratio between the acetoacetate and the beta-hydroxybutyrate? And those questions are all on the table, and people need to do some work on that. We're taking some steps in that direction as well. I'll be mentioning a little bit about that in my talk. But these are things we need to solve. Should we be giving the ketones directly preformed, or should we not be? Should we be using the fatty acids in our fat stores and in the diet through a ketogenic diet as opposed to MCT? Is that going to give us a better ratio and a more sustained delivery of ketones? Because on a ketogenic diet, you're on it 24 hours a day. Even when you're not eating, of course, you're in. Whereas the MCT are burned very quickly, and so you have highs and lows depending on how many times you take it per day. So it's a different physiological response, and it might be important to know whether one or the other is advantageous, and maybe we'll find out that they're equivalent. Sort of like in epilepsy treatment, the MCTs seem to be broadly similar in effect to the ketogenic diet. So, But does that mean that's the case in Alzheimer's? I mean, we just don't know